individual people have a different distribution of the three muscle fiber types that we've discussed: the slow oxidative or type 1 fibers, the fast oxidative or type 2A fibers, and the fast glycolytic or type 2B fibers. Different people have different percentages of each one of those types of fibers in their muscles, and the reason it varies from person to person is because people inherit that distribution largely from their parents, from their ancestors. On this slide, you're looking at some images that were taken under a light microscope. These are images of cross sections through a fascicle of a muscle. So remember, a fascicle is a combination of many individual muscle fibers, muscle cells, that are all joined together. And since we've cut that fascicle in the transverse plane, since we've created a cross section, when we look at that slide, each one of these individual structures that you can see outlined here, each one of these is one muscle fiber, one muscle cell in cross section. We're looking at the cut face. Now, we have the ability to stain type 1 cells specifically, or to stain type 2 cells specifically, or to stain type 2A versus type 2B cells. But it requires, different types of stains to see those different categories of muscle fibers. This image up at the top here has been stained to show where the type 1 fibers are, the slow oxidative fibers are. And they are located everywhere you see this dark, sort of purple blue staining. Those are all type 1 fibers, again, in cross section. The other fibers, the ones that did not pick up stain, are the fast type fibers. The fast oxidative, that's type 2A, and fast glycolytic, that's type 2B. You would have to use a different stain in order to distinguish those from each other. Now, just by eyeballing this looks like there's approximately half of these fibers falling into the type 1 category. So about 50% of these fibers are type 1, are slow oxidative. Now, if you look at the image in the middle, this is another slide that was prepared, and this time type 2 fibers were stained. Now, they did not stain to differentiate the type 2A from the type 2B. So everywhere you see the purple stain, those are just type 2. Some of them are type 2A and some of them are type 2B. Again, you might say about 50-50. About half of the cells picked up the purple stain and half didn't. So about half of these are type 2, but we don't know what percentage in there is type 2A and what's type 2B. So we have different staining abilities, and we can stain for different materials in a cell and tell what characteristics those cells have. Down here, this is what an unstained preparation looks like. So again, they made slides, and before they were stained, it would have looked like this. Now, we've said that the sarcomere is the fundamental unit of contraction in skeletal muscle. We've said that the way the sarcomere performs its function is by having the thick filaments made out of myosin bind to and pull on the thin filaments, sliding them past each other in order to shorten the sarcomere. you can imagine that in order for sarcomeres to function correctly, they have to maintain that very precise geometry that they have, that hexagonal pattern of thin filaments arranged very precisely around each thick filament. So the question then is, how do sarcomeres stay in such perfect geometrical patterning? Why don't they sort of, bend and twist and fall out of shape? And the answer is that they are loaded with proteins that help them maintain their shape, help keep the thick and the thin filaments separated from each other in precise organization. Remember, the thin filaments are anchored at the Z lines. The thick filaments are anchored at the M line. The thick filaments, remember, as they grab onto the thin filaments and slide them past each other, they are going to actually appear as though they are moving toward the Z lines as the sarcomere gets shorter and shorter and shorter. The thick filaments themselves are staying put. It's the thin filaments that are moving. And as those thin filaments move towards each other, as the Z lines get closer and closer and closer together, it will appear like the thick filaments are moving towards the Z lines. In addition to the thick and thin filaments, there are other structural proteins. Yes, troponin and tropomyosin, those are the two that are laying on the thin filaments. But there are also structural proteins that are placed to help keep the thin filaments exactly spaced apart from each other and keep them exactly parallel to each other. On this slide, you can see just a couple of those accessory proteins. Over here on the right, we're looking at about half of a sarcomere. Here's a Z line, and here are some blue thin filaments that are attached to, anchored to that Z line and protruding off at a 90-degree angle. Now, the thick filaments are shown in brown here. And right away we can see that there are some other proteins in here. For example, there's this sort of brownish colored protein, this line of brown lying just underneath that thin filament. There's a protein shown in green right here, sort of capping off the end of that thin filament. There's a protein shown in this gray color on both sides of the Z line. There's a protein sort of wrapped around the very center of the thick filament. There are all kinds of accessory proteins in here that help maintain this geometry and help keep all of these filaments perfectly aligned with each other so that it can function correctly. Because during contraction, these brown thick filaments are going to be attaching to the thin filaments and pulling on them. On this side, the thin filaments are going to move this way, this way. On this side, the thin filaments are going to move this way. So these two Z lines are going to get closer and closer and closer to each other during contraction. The length of the sarcomere, in other words, is going to shorten during contraction. This area right here, this I band area, is going to go away because the Z line is going to move this way, this way, this way, until it essentially bumps into the thick filaments. Over here, this Z line is going to go this way until it bumps into those thick filaments. Once the sarcomere is completely contracted, the Z lines will lie right here and right here. That sarcomere will have gotten as short as it can get. And that brings us to what we call the sliding filament model of contraction. This is the physiology of muscle contraction. And as we've said, it involves these thick and thin filaments actually sliding past each other. The thick filaments grab onto the thin filaments, the myosin heads bind to the thin filaments and slide them towards each other in the middle of the sarcomere. Those heads on the myosins in the thick filament are going to pull on the thin filaments using the energy that comes from ATP. The thin filament is going to slide. Remember, the thick filament has mirror halves. So one half of the thick filament is going to pull thin filaments in one direction towards the center, and the other half is going to pull thin filaments in the other direction towards the center. And we're going to shorten that sarcomere. So neither filament is physically getting shorter. The sarcomere is getting shorter. The filaments themselves are staying the same length, but the Z lines are coming closer and closer together, so the sarcomere is getting shorter. If you watch underneath the microscope, the A bands will actually stay the same length during contraction. It's the I bands that get shorter and shorter and ultimately disappear. So sarcomeres get shorter. That causes the myofibrils to get shorter. That causes the muscle fibers to get shorter, which causes the fascicles to get shorter, which causes the muscle to get shorter, to contract. The process of muscle contraction generates force. This is a force that allows for the movement of the bones in the skeleton, and it allows for individual people to physically move. Now, that force that gets generated through the shortening of sarcomeres is actually transferred to all those fibrous coverings around muscle. Remember, we talked about the endomysium and the perimysium and the epimysium and then the fascia and then the tendons. The force gets transferred through those fibrous coverings, essentially pulling on the bones and helping the skeleton to move. Now, this force that gets generated in muscle, this has to be strong enough to overcome any resistance in those structures. And once the force is greater than the resistance, then you get that movement of not only the muscle, but the skeleton that the muscle is attached to. This drawing over here on the right, I really like because it reminds us that sarcomeres are not two-dimensional structures, they are three-dimensional structures. We're not looking at an entire sarcomere here because there's only one thick filament in our view. But this thick filament is surrounded in 360 degrees by thin filaments, shown in brown. And this is that honeycombing pattern, at least part of it, that we see. So in a relaxed state, you'll notice that this sarcomere is relatively wide. There is overlap, there's always overlap between the thin and the thick filaments, where the heads are located on either side. There's always some overlap, but we've got a nice, open, relaxed sarcomere here. We can see the whole A band. We can see both I bands. Now, during contraction, the sarcomere gets shorter. These globular heads on either side of the thick filament are attaching to and sliding these thin filaments towards the center. This side is being slid in this direction. This side is being moved in this direction. so that the ends of the sarcomere, the Z lines, are getting closer and closer to each other. The I bands will eventually disappear because all of the thin and thick filaments are now overlapped. It's not that the thin filaments are getting shorter. It's not that the thick filament is getting shorter. It's the sarcomere that's getting shorter because the thick filament is sliding all of the thin filaments closer together. Now, it's important to note that there is a limit to muscle contraction, and that's because there is a limit to the anatomy of a sarcomere. Take a look at the drawing on the left side of the slide here. Up at the top, you're looking at two sarcomeres side by side. Here's a Z line, and here's a Z line. So this is one sarcomere. This Z line to this Z line, that's the other sarcomere. We can see thick filaments in brown, thin filaments in blue. While the sarcomeres are relaxed, there's some overlap between the thick and the thin. We can see an A band here, an I band here, and an I band here. Now, during contraction, these two sarcomeres are going to shorten. The thick filaments are going to bind to and slide the thin filaments, shortening the length of each one of these sarcomeres. That shortening can continue until the Z lines come in contact with the thick filaments, essentially. There's some of the accessory protein in here preventing them from literally crushing each other, but the Z lines are going to keep getting closer and closer and closer to where the thick filaments are until they hit that point. At this time, these sarcomeres are maximally contracted. If you were to push this contraction past this point, you would damage the sarcomere. Because if you tried to shorten this sarcomere anymore, you wouldn't be able to slide the thin filaments. You would literally just have to crush the Z line into the thick filaments, and that would damage the sarcomere. So that's why there is a limit to muscle contraction and the force that is generated in muscle contraction. And that's what generates this graph over here. This is called a length tension curve. It draws a comparison between the length of the sarcomere and the tension or the force that can be generated by shortening a sarcomere. Up at the top, we're looking at a sarcomere in its relaxed state, into a contracted state, into a maximally contracted state, and then into an over-contracted state, essentially. So in order to read this diagram correctly, you actually have to start over here. So the x-axis is showing us the length of the sarcomere, and of course, we're at a microscopic level, so we're at micrometers here. These are very small structures. And then on the x-axis, we're looking at the tension or the force that gets generated through the contraction of a sarcomere. So if you start over here, where you have zero force generated, the sarcomere is at its most relaxed point. And that's what they're showing us at the top here. There's always a little bit of overlap. There are a few heads, a few globular heads overlapped with thin filaments on either side in a relaxed sarcomere. Now, if I start to shorten the sarcomere, if I start to make it shorter and shorter, heading this way on the x-axis, I'm also going to start generating force. The red line goes up, 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 up as the sarcomere gets shorter and shorter and shorter. But then we're going to hit a plateau. We're going to generate the maximum force that can be generated once the sarcomere is fully contracted. You can keep trying past that point, you can keep trying to shorten the sarcomere, but if you go past that maximal overlap, that maximal contraction point, this happens. Force drops off, and then it plummets. So what the length tension curve shows us is that there is a maximal point of contraction and a maximum force that is generated in a muscle. If you try to push it past that, you will damage the sarcomere and you will lose power in the muscle as a result. Now, there is another term that we should be familiar with here, and it's a term called recruitment. We've said that there's a limit to how far an individual sarcomere can contract. And that means that there's a limit to how many sarcomeres within the myofibril can be maximally contracted. And that means that all of the myofibrils within a muscle cell, a muscle fiber, can all become maximally contracted. Now, this word recruitment refers to the fact that when a person tries to use a skeletal muscle, the entirety of a skeletal muscle, again, I'll use the example of the biceps brachii, when you're trying to contract your biceps brachii muscle, you can get a terrific level of fine tuning to that contraction through recruitment, because you can add or subtract the number of individual muscle fibers that you are asking to contract at any one time. So, in other words, yes, there is a maximum level of contraction for each individual sarcomere, but because there are so many individual muscle fibers, individual cells inside a muscle, that means that depending on how many of those fibers have been recruited, have joined in to the effort of contraction, that's going to determine just how contracted or relaxed that muscle is. So it's not an all or none. Muscle contraction has a tremendous amount of variability in it because of the fact that the act of contraction can involve as many or as few muscle fibers as needed, and everything in between. Depending upon the number of muscle fibers that are brought in, that are recruited, that's going to impact the ultimate combined force of the contraction of that muscle. Let's go back now and talk about the sliding filament model of contraction, and specifically how these cross bridges form. It's the myosin heads that do the actual physical shape changing using the energy of ATP, that are binding to the thin filaments and causing the thin filaments to slide past the thick filaments. Every time a myosin head binds to a thin filament, it creates what we call a cross bridge. It's transient binding, though, because the sliding has to continue, right? The head, in other words, has to bind to the thin filament, slide it a little bit, then it has to let go. It has to bind to the next spot on the thin filament and slide a little more. And then it has to let go and bind to the next spot and slide. So these cross bridges that form between the thick and thin filaments, these are transient structures. They form and then they let go, and then they form and then they let go. And every time a cross bridge forms, it forms because a molecule of ATP has been broken down. And remember, on the globular head, there's a there's a region that acts like an enzyme. It's catalytic. And what it's doing is breaking apart ATP. When the myosin head breaks an ATP molecule, it uses that energy to change shape, and it binds to the thin filament and it slides it. That sliding motion we refer to as a power stroke. Then it releases the thin filament. Then it binds to another spot. It turns out that the release of the myosin head from the thin filament also requires a shape change. That makes sense, right? And that means another ATP has to bind and be broken so that the myosin can let go. So throughout these motions that the myosin heads are doing, throughout these sliding motions that are going on during contraction, we're going to see two ATP molecules attaching and being broken and providing the energy needed for contraction. When you watch a sarcomere through the process of contraction, there are really 4 steps to it. The myosin molecule, the globular heads, are going to go through a shape change. They're going to essentially cock backwards. And then they're going to move upwards, bind to the thin filament, and then push the thin filament, slide the thin filament. That's that power stroke. And then they're going to let go. And then the whole thing repeats. They cock backwards, those heads. They bind to the thin filament, power stroke, slide it, and then let go. ATP is required at 2 points. You need a molecule of ATP to bind in order for that myosin head to cock backwards. And then you need another ATP to bind when it's time for the myosin head to let go. A lot of ATP is required for muscle contraction. I'm going to add one more thing here, and it has to do with ATP. Remember, when we've talked about ATP in the past, we said that cells do not store significant amounts of ATP. Instead, they make it as they need it. And that's why, for example, if a human being stops breathing, they only have a few minutes before death comes because they don't have any fresh oxygen coming in and they can't make any fresh ATP. We only have a couple minutes worth of ATP at any one moment. So what muscle fibers have done through evolution to try to get around this problem? Well, really, they've done 2 things. Remember, myoglobin is present in muscle fibers, and myoglobin can store some extra oxygen so that should there be a sudden disruption in oxygen supply, the myoglobin can provide at least a little bit of oxygen in that moment that can be used in the cellular respiration and oxidative phosphorylation process to make some ATP. The other thing that muscle fibers have evolved to be able to do is use a molecule called creatine phosphate. So what happens is ATP is able to donate, if you will, a phosphate group to this molecule called creatine or creatine. And when it does that, it creates a molecule called creatine phosphate. Now, that phosphate group is essentially being stored by attaching it to the creatine molecule. Now, when contraction begins and the cell needs ATP in order for contraction to occur, the cell can reach to that creatine phosphate, ask it to donate the phosphate group back, essentially. An enzyme does that work. Take the phosphate back off of the creatine, and make more ATP in that moment by putting the phosphate group back on to the ADP that's present in the cell. And now you have more ATP. So by taking it off of the ATP and putting it onto the creatine, it looks like there's actually less ATP around. There's more ADP now and less ATP. So you get around this problem of not being able to store a lot of ATP by taking the phosphate group off and putting it onto creatine. Creatine phosphate is there in a pinch to donate a phosphate group to ADP and make ATP in a muscle fiber. On this slide, we can see the role that creatine and creatine phosphate play in a muscle fiber. If you look over on the left side, we're looking at the state of relaxed muscle. And over on the right, we're looking at the state of contracting muscle. So if you start at the left, this muscle is relaxed. It doesn't have a need for a lot of ATP right now. There's plenty of it around. It's just not using it in this current moment. So this ATP can be converted into ADP and a phosphate group. And the phosphate group can be given to this molecule, the creatine, creating creatine phosphate or creatine phosphate. Now, when the muscle needs to start contracting, what will happen is an enzyme will take that phosphate group back off of the creatine phosphate molecule and give it to ADP, creating ATP. In other words, in a moment's notice, essentially, we can have more ATP available. Now, you might say to yourself, well, why not just leave the ATP alone? Why not just have the ATP sit there? Well, remember, ATP doesn't get stored in a cell. So this ATP, if it's not going to be used in a relaxing muscle, it's going to get pulled away and used for something else. But by donating its phosphate group to the creatine, it's as though it is being stored, at least a little of it. You get about 15 seconds worth from your creatine phosphate stores in a muscle fiber.